Okay. Yeah, so I'm really happy to finally connect with this group. I've been talking to Katie for a while, and I tried like hell to get out there to join you all this week, but administrative duties prevailed, unfortunately. So some administrative duties you just can't shirk, I imagine. So I think Jenna or Katie's going to control my slides, so if we want to go to the next slide. So I just want to give a quick background about NIST. So I know some people are familiar with NIST, some are not. So most developed countries have a national metrology institute that deals with standards of measurements. Of course, in the United States, NIST is that metrology institute. So we are a non-regulatory federal government agency in the Department of Commerce. We have about 3,000 employees, which are mainly scientists and engineers, typically physicists, engineers, chemists, etc. About 2,800 associates and facility users. And about 400 NIST staff are on about 1,000 national and international standards committees. So we're located in Gaithersburg, Maryland. That's the main campus. I haven't found an aerial photo yet that accurately captures our campus, but we're about 62 buildings, so we're rather large. And we also have a campus in Boulder, Colorado, and a few other joint institutes around the country, including Stanford, Charleston, South Carolina, etc. But you may be asking, why is a scientific technical institute in the Department of Commerce? And it's because industry is our stakeholder. NIST vision or mission is to be the world's leader in creating critical measurement solutions and promoting equitable standards. Our efforts stimulate innovation, foster industrial competitiveness, and improve quality of life. So that's just a quick 101 background of NIST. The next slide. So I want to tell you a quick story about why it is that we do what we do in the biosciences. So we could talk all day about the atomic clocks and the meter and the kilogram, etc. But specifically, we have a bioscience program here that focuses on bioscience-related issues. So a quick story on why we do what we do. So as I'm sure you all know, back in 2013, the FDA, for the first time, approved a next-gen sequencing instrument for clinical diagnostic use. Of course, we know now that that's the Illumina MiSeq instrument, or specifically the MiSeq DX. So to sort of celebrate this new time in history, Francis Collins and Margaret Hamburg, respectively the heads of NIH and FDA, wrote a piece, perspective, in the New England Journal of Medicine, and it was highlighting this transformative time in clinical diagnostics, that is the introduction of next-gen sequencing. And in this article, they mentioned that contingent on the regulatory approval of this new diagnostic instrument was the development of standards and reference materials that allow us to assess the accuracy of human genetic testing. And they specifically mentioned that NIST was going to collaborate with FDA to develop these standards. So this really kicked off our Human Genome in a Bottle program, which has been a wildly successful program for the last few years. And if we go to the next slide, I'll just highlight it real quickly. It is exactly what it sounds like it is. It's human genomic DNA in a tube. It's probably the most well-characterized human genome in the world. We have not only the DNA that's made available to our stakeholders, but obviously the reference data that goes along with it and the bioinformatic tools that allow you to make sense of that data. So one can buy the DNA, sequence it in your own lab, use your own bioinformatic tools to make variant calls, etc., and then compare and see how well you did. And I'm sure some of you are also familiar with the precision FDA movement that has started up recently, and a lot of this is based on the availability of this NIST human genomic reference material. And next slide. This was recently highlighted in the New York Times, so I think this is a very succinct, accurate statement that just really describes what we do. U.S. introduces new DNA standard for ensuring accuracy of genetic tests. So that's just a very clear, straightforward example of how NIST develops standards in measurement science to help human health in the clinical diagnostic arena. Next slide. So this slide really just captures what I just mentioned. So we're a non-regulatory agency. FDA is the regulatory agency. So we partner directly with industry, and we partner directly with FDA. And I really think this slide captures the relationship that, you know, FDA often funds us to develop reference materials that then industry adopts for regulatory issues. So it's a real synergy and interplay between these two agencies and our stakeholders who are industry. Next slide. Another example of a reference material we made for the bioscience community was, and this was actually something that came out quite a while back, RNA spike-ins to assess the sensitivity and specificity of microarray-based gene expression experiments. So we started this project 10 years ago, specifically Mark Sallet and Sarah Monroe. At the time, microarray technology was the dominant genomic technology, and I have to say that obviously we've seen a lot of that shift to next-gen sequencing RNA-seq-based measurements, but these standards still apply just as applicably. So as you can see, we have reference materials available, and Ambien has even sort of developed their own version of our ERCC material that you can buy commercially from them, and it's a little more convenient to use because they've got it mixed and spiked, et cetera. That just makes it a little easier for the end user to use. So again, it's an RNA spiking material to assess the sensitivity and specificity of gene expression experiments. Next slide. So when we think of regulatory human health, we think of clinical diagnostics, and we also think of human therapeutics. And when people think about safety and efficacy of human therapeutics, you often think about clinical trials and cohort studies, et cetera. But I would argue that safety and efficacy of a new therapeutic can be demonstrated in the lab via innovative measurements as well. Specifically, those measurements are focused around demonstrating purity, stability, identity, and potency of therapeutics. So one of the larger, more successful programs here in the Material Measurement Laboratory is our biomanufacturing program. So when I say biomanufacturing, I'm referring to the use of cells, whether they're bacterial cells or CHO cells, to produce high-value molecules. And in the case of biomanufacturing therapeutics, those are typically protein-based therapeutics. So we partner with pharma industry to develop the new measurements that ensure purity, identity, stability, and potency of protein-based therapeutics. And this falls in line with the FDA's Chemical Manufacturing Controls Program that essentially regulates how these drugs are manufactured in a safe way. So our program is large. We collaborate with many pharma companies. We spend about $10 million of our own budget annually on developing novel measurements to characterize these new protein-based therapeutics. And we also have about 5 joint postdocs between ourselves and pharma companies. So next slide. Just an example of how we're serving this industry, not only developing the measurements that help identify what these therapeutics are, potency, purity, et cetera. But we recently came out with this, we call it the NIST Map. It's a NIST monoclonal antibody. So as you probably know, most protein-based therapeutics are monoclonal antibodies, large biomolecules. And industry needs a pre-competitive resource to allow them to develop new analytical methods to ensure quality control. So now they have a pre-competitive tool that measurements can be applied to, and information can be shared for the mutual gain of the entire industry, and to help FDA make regulatory decisions. Next slide. And I like to think, so the statistic that people throw out a lot is that in 1983 there was one protein-based therapeutic, and that was human insulin. 30 years later, current day, 7 of the top 10 selling drugs are proteins. So we're now beginning to see a new class of therapeutics emerge. Those are microbes, so obviously microbiome-targeted therapeutics, whether the therapeutic is a microbe or whether it's a small molecule prebiotic that targets the microbiome. Either way, we're dealing with a new class of therapeutics, and this is just a list of some pharma companies that have popped up recently that are devoted to developing microbiome therapeutics. So just as industry and NIST has historically provided measurements to demonstrate purity, stability, identity, and potency of protein-based therapeutics, we're entering a brand new world of therapeutic measurements. So how does one begin to demonstrate purity of a microbiome therapeutic? If I have a pill that is a mixture of bacteria and I'm asked to demonstrate the purity of it, I'm not even sure what that definition is. Are we talking about genetic purity, or are we talking about chemical metabolite purity? If it's genetic purity, to what level, what standard of purity? Are SNPs that are introduced into genomes during the culturing of these bacteria considered impurities? So these are regulatory questions that have to be figured out to some extent, and NIST historically has played a role in doing this. Next slide. So the microbial metrology program at NIST is relatively new, so there's always been some microbiology going on at NIST, but just in the last 2 years, NIST decided that they really wanted to stake a flag in the ground, so to speak, and develop both a 5-year strategic plan in microbial metrology as well as establishing a new group in microbial metrology and really formalized microbiology at NIST. And our strategic plan as it currently stands is really focused on 5 main areas, which I consider each to be sort of an independent program. And those 5 programs are microbiome community measurements, which I'm here to talk to you about today, going counterclockwise, combating antimicrobial resistance. So we have some novel, innovative ways of looking at the emergence of resistance using microfluidic and microfluidic tools to monitor real-time emergence of antibiotic resistance and ergo bacterial evolution. Pathogen detection and identification, so we've got funding from the FDA to develop standards for pathogen detection via next-gen sequencing, and this is to support the clinical diagnostic community. Biofilms and engineering biology. So I'm just going to take the next 5 or 10 minutes to talk to you about our microbiome community measurements, which is, again, what I wanted to share with you all today. So next slide. So I think this article captures the problem very well. This article is 2 years old, but not too much has changed. So some of you may have seen this article in Science News by Tina Say a couple of years ago. I think it came out in 2014. So essentially what she had done in this article is she took a sample of her own gut microbiome, a poop sample. She split it in half, and she packaged it up, and she sent it to 2 services that provide gut microbiome analysis for fees, so American Gut and MuBiome. She got the results back a couple weeks later, and what she found is that the results were almost entirely opposite. So different results from the 2 different companies. And she did her due diligence and went to the leaders of these companies and told what had happened, and she wrote a very nice article on it. And it's not to poo-poo, no pun intended, not to poo-poo what anyone has done. It's just an indicator of the state of this new field. So some of the quotes in this article that I thought were very NIST-y, the lack of reproducibility between studies was frustrating. To me, it seemed like cowboy country. It needed to have some kind of order. I think Rob Knight was quoted in here. All sorts of unlikely things are possible, and finding out which one is true is difficult. And finally, scientists are trying to find ways to standardize microbiome studies so that they can directly compare the results. Okay, so this was a nice, somewhat tongue-in-cheek piece about, the need for improved microbiome measurements and understanding sources of bias, etc. Next slide. So how do we think about uncertainty in a microbiome measurement? So you have a starting sample. Your starting sample may be a stool sample. It may be a skin sample. It may be a built environment sample. So how you handle that sample is ultimately going to affect your results. And I'm talking specifically about the isolation of your sample, how you stabilize it if you use a chemical stabilization or freezing, how it's stored, how it's shipped, etc. So all of these things will ultimately affect the final result. Then, of course, maybe arguably the largest source of bias is DNA extraction. So DNA extraction efficiency is obviously a very mechanical process. The way one person does it may be different than another. There are inherent biases in these. Some methods are better at extracting gram-positive. Some are better at gram-negatives, et cetera, et cetera. And the mechanical processes themselves, whether they're done manually by hand or automated in a liquid handling instrument, you can imagine all sorts of biases may be introduced. And then once you have extracted purified DNA, you have options regarding how you're going to characterize it, whether it's a 16S rDNA PCR assay, where you're just looking at the 16S as a taxonomic identifier, or next-gen, whoa, my computer just froze. Okay, I'm back. Or whether you're doing a shotgun next-gen sequencing approach, where obviously you sequence all the DNA. So from the 16S PCR approach, your selection of primers, which variable regions you're looking at, how long you're sequencing, et cetera, the number of PCR cycles, et cetera. And on the shotgun next-gen sequencing library prep side, you have obviously different library prep tools, anything from the Nextera kits to the other sequencing platforms like the Ion Torrent that I have shown here or others. And moving on to the next-gen sequencing platform, different platforms have inherent biases associated with them, and we've shown that, and I'm sure others have seen that as well. And finally, to bioinformatics And then one of the things that was highlighted in the Science News article that I mentioned in the last slide is that different bioinformaticists looking at the same raw data set may get different results based on how they do their analysis and how they interpret the data. Okay, next slide. So having said that, sort of setting the stage, this all sort of came to a head or came to fruition in 2015 during the Fast Track Action Committee mapping the microbiome. I'm sure all of you in the room are familiar with this. The Fast Track Action Committee out of the Office of Science and Technology Policy. So the report came out in November 2015. You all know, I'm sure, the purpose of the Fast Track was to identify areas of current federal investment, research needs, and resource gaps. So not only was it an audit of what research had happened in the last 3 years, but also an assessment of what's needed to move forward, to translate basic research into clinical applications, agricultural applications, health applications, et cetera. So next slide. If you all saw the report in the Appendix 15, so I'm looking at the big blue bar graph on the right, and I'm sorry it looks fuzzy, but these are expressed needs or research gaps, things that need to be fixed in order for us to move forward, to move this powerful new field forward translated into applications. And the bar represents sort of the strength of the need or the number of people in the committee who recognize the need. And the first at the very bottom is computational modeling skills, bioinformatics, et cetera, which is an obvious one. But second to that was the identification of the needs of reference materials, standards, et cetera. And this wasn't, I was the NIST representative on this action committee, but it wasn't just my voice at the table. It was nearly unanimously recognized that there needs to be standards and reference materials introduced in order for us to take this field of research to the next stage. Next slide. So what does NIST typically do when we commit to a new program, something like microbiome measurements? So we are very stakeholder-driven. We are not regulatory. We do not impose our standards. They are adopted through community consent. They're often defined by community consent. So what we typically do when we commit to a new area such as this is we host a workshop, and we bring in our stakeholders and we say, tell us what you need, and we will, tell us what you need, and we'll help you get there. So back in August 9th and 10th, just a few months ago, NIST partnered with NIH, and we hosted the Standards for Microbiome Measurements workshop. It was a 2-day workshop. The pie chart on the bottom left shows the breakdown of the attendees. So we had a little over 200 attendees. Most of those were from government, FDA, NIH, CDC, USDA, et cetera. A number of attendees were from academics, industry, military, not-profit, and I think we had about 26 international attendees. And some of those international attendees were from other metrology institutes at other developed countries. All of this, so I could give you, we're writing a report on this, so we will publish a workshop report. The entire 2-day workshop was videotaped, and the entire webcast is available on our website. So if you Google NIST microbiome, when I did it this morning, it was the first hit that came up with our Standards for Microbiome Measurements workshop website. The video is right there. I think it's 10 or 12 hours of edited video. So I think that picture is Matt Henn from Series Therapeutics talking about their microbiome therapeutic pipeline. And I will say, too, because I'm talking to representatives from the built environment, that while we recognize that there are not only different microbiome or microbial ecosystems, we also recognize there are different types of measurements that you can do on those. So DNA measurements, of course, are the fundamental measurements, but you can also do mass spec-based metabolomics and mass spec-based glycomics and lipidomics, et cetera. So in thinking about, everything that we can focus on during this workshop, we decided it would behoove us and it would benefit everyone if we really narrowed down the focus to a specific thing that we can tackle. And to me, it was the thing that's the lowest hanging fruit, and that is microbiome therapeutics and diagnostics for clinical applications. So we specifically focused the workshop, the speakers, the discussion, et cetera, on DNA-based measurements of the human microbiome, specifically for therapeutics and diagnostics. So this is workshop number 1 of what I hope to be an annual event. As we press forward, we can begin to talk about other measurements and even other ecosystems, et cetera. Next slide. When we were planning the microbiome standards workshop, I was getting calls from various people in the field, and one of which expressed interest was the Phytobiome Society. So, of course, these are the folks that study the soil microbes and all organisms related to plants in order to try to improve agricultural yields, agricultural processes, et cetera. So think of soil probiotics for enhancing crop yields, et cetera. So they asked if they could have sort of a mini workshop here with us at NIST to discuss the agricultural specific needs of microbiome measurements. So the same week that we had the larger microbiome workshop, the 9th and the 10th, on the 12th of that week, which was the Friday, we hosted the Phytobiome Society. It was a smaller workshop, maybe 35 or 40 people. And we focused the entire day on short presentations and breakout sessions and discussions and brainstorming on what measurement needs are unique to the agricultural community, to the phytobiome community, and how can we address those. And also, as we all know, while some of the samples that are collected and how they're collected and how they're stored are unique to different ecosystems, whether it's soil or skin or stool or the International Space Station, the measurement process afterwards is roughly the same. So the same principles can be applied no matter what ecosystem you're really looking at, which is one of the reasons why I was looking forward to connecting to the built environment folks to really discuss the sample-specific needs, et cetera, that maybe differ from human stool and skin, et cetera. Next slide. I also want to point out, and this is something I should have mentioned earlier, we recognize that other microbiome and metagenomic standards consortia exist. So Rob just mentioned one in his previous talk, perhaps the one that's done the most towards understanding bias, et cetera, is the Microbiome Quality Control Consortia project, and I know they put out a couple papers recently. We really use this as our starting point, our jumping off point. We're certainly not here to duplicate efforts. We're not here to step on toes. We want to learn as much as we can from what's been done historically, and we want to build from it and begin to make contributions towards it. So in addition to the MBQC, there's also the International Human Microbiome Standards Project, which was represented by a speaker at our standards workshop. Again, these were the first people we called to say, come in and talk to us about what you've done in this field. The Association of Biomolecular Resource Facilities, ABRF, their Genomics Research Group, MGRG, which is chaired by Scott Ty at University of Vermont, and the Genomic Standards Consortia. And they're the folks that are the editors of the journal Standards in Genomics. So, yeah. So next slide. So I mentioned that we recognize there's a half a dozen standards consortia that exist. So we said, hey, let's do it again. So actually, it was kind of a roundabout story. So just through phone calls and conversations with people in the field and academics and industry, et cetera, who were interested in improving microbiome measurements, understanding bias, we began having weekly, bi-weekly phone calls just to discuss common interests and try to cross-fertilize ourselves with, what's going on in individual labs. And so-and-so just made a new mock community in their lab, and they're sharing it with people. And, here's what we did with a quick little interlab study. And we decided that we could sort of, by hosting this out of NIST, we could sort of be the glue that brings together industry, international metrology institutes, previous standards consortia, et cetera, to really harness all that's been done previously and begin to build from it and understand where we need to go next. So IMSA, as I affectionately refer to it, is the International Metagenomics and Microbiome Standards Alliance. And if you Google IMSA, Google IMSA and NIST, you'll find us on a NIST website. It has our mission statement. I'll just read briefly. We're a non-hierarchical association of microbiome-focused researchers from industry, academia, and government. We were formed for the mutual benefit of the entire microbiome and metagenomics community. We're not ecosystem specific. We're not technology or measurement specific. So whether it's genomics, epidemiology, forensics, bioinformatics, statistics, human environment, built environment, it doesn't matter. If you're interested in measurement assurance regarding microbiome measurements, we would love to have you. So it's an open alliance. Again, if you contact me, we'll be happy to get you on the monthly WebEx. And it's not just discussions anymore. We're going to begin to push forward on development of interlab studies, and I'm sure Rob mentioned that the MBQC is starting up their MBQC 2.0, as I call it, and it's going to be a brand new round of interlab studies, and we've got brand new mock communities and brand new bioinformatic tools. So we certainly want to be part of this and contribute to this. Next slide. I may have just captured this last slide in my last sentence. So one of the big things that people look at this and they say, are you going to develop a reference material? So what does a microbiome reference material look like? So, the most obvious intuitive thing is a mock community, so an engineered community that serves as ground truth. So we know exactly what it is before you measure it. Is this something NIST is going to do? We certainly aren't opposed to the idea. I mean, this is what we're here for. So if this is what the community wants, we'll do it. My biggest pushback right now is that and I would like to do something like this, but I will say that my biggest pushback is that I happen to know of a half a dozen of mock communities that are available currently that aren't being adopted by the community. So almost sarcastically, I ask, why would we develop the seventh one that people don't use? So the reason we exist is to serve this community, and we will do whatever is needed to serve our purpose. But we're still deciding exactly what sort of reference material standards are needed by the community. So we have expertise in genomic metrology, so as I mentioned, the RNA spike-ins to the human genomic reference material, et cetera, et cetera. So we have years of experience in genomic metrology, understanding biases associated with next-gen sequencing instruments, et cetera. Transcriptomics, we have a lot of mass spec metabolomic expertise here. So I will say that no one here yet is doing microbiome metabolomics, but we do have a lot of mass spec metabolomic expertise because two reference materials that NIST has and has had for many, many years is a human blood reference material and a human urine reference material. And our metabolomics gurus are downstairs right now characterizing that reference material using metabolomic approaches. So obviously, blood and urine is used in a clinical diagnostic setting as a reference material for sensitivity, specificity, et cetera. So we need to characterize what metabolites are in those materials before we can validate them and certify them, et cetera. Also, cell counting and bioinformatics. So that's sort of the technique, the technical expertise we have here. We have lots of physicists and engineers developing microfluidic tools, et cetera. So another project that we're spinning up is what we call the engineered communities environment. So we're going to begin using 2D and 3D printing technology to print microscopic microbial communities. So think of single cell printing in microscopic petri dishes and then using hyperspectral imaging and other techniques to really understand what defines community resilience. Community engagement. So I said that's one thing that NIST does well. So we can bring people together. So the international metrology efforts to develop other standards in other countries for microbiome and metagenomics efforts, and there are other metrology agencies in the world that are working on this problem right now quite prolifically. We can bring together industry, government, academic partners, international partners, et cetera. Next slide. We do have postdoc opportunities available. If anyone, if you all have PhD students that are looking for postdocs, NIST is a great place to work. We jokingly say that people leave NIST horizontally because once you come in, you typically don't leave until you absolutely have to leave. So they're well-paying, NRC-funded postdocs. So if any potential students point this way. Next slide. And that's all. And again, I really appreciate you all allowing me to connect with this community. It's been something I've been trying to do for about a year now, and glad it finally came to be.
Thanks, Scott. I think
so. I'll take any questions.
There's probably time for just one or two quick questions to try and keep on track. So.
Okay.
So I'll ask a quick question, Scott. You mentioned that you're trying to decide what kind of reference material that you want to sort of, whether reference material is actually of use right now. Do you have a sort of a sense of where in that chain that you would like to focus on? Is it primarily starting at the DNA, point of DNA, and collecting sets of DNA reference material, or are there other parts where you see a gap right now that are maybe less well sort of populated?
Yeah, so engineering microbial communities at the cellular level, mock communities, those are notoriously difficult to do well because, while it may not seem is the case, but it's very difficult to accurately count cells. And, bacterial cells form clumps and filaments, et cetera, so it's really difficult to make. And in fact, that's really why what sparked us to develop this 2D printing tool as a way to make defined microbial communities that we can study. Yes, the development of mock communities is something that we certainly feel is necessary. It's the most intuitive, straightforward way of assessing ground truth. DNA mixtures is the next step forward. Now, of course, a DNA-based reference material that is a representative of a diverse community wouldn't assess the biases associated with DNA extraction. You sort of skip over that step, but nevertheless, it can be a very important, informative, valuable tool. And then moving one step past that is standard reference data, and we do have a new Office of Data Informatics, and we do have standard reference data sets. So all of the above, cell-based reference material, DNA-based reference materials, and bioinformatic data sets.
I guess one.
Do you have any experience including fungi in your mock communities? So the question was, do you have any experience including fungi in your mock communities? Standard communities.
So we have acted as an advisor on the development of a couple other mock communities outside of NIST, and those have always included fungi, specifically Saccharomyces cerevisiae. So Saccharomyces cerevisiae, of course, is a tough nut to crack. So it's a good one to include in controls. Of course, it doesn't really get included when you do 16S-based measurements. That's a different story. But yes, we recognize not only the importance of fungi in microbiomes, but also in the reference materials. But of course, because of course fungi are very relevant and important in the health of human microbiomes and infectious disease, et cetera. So in moving forward, we would certainly include, a diversity of organisms. And yes.
Okay, thanks, Scott. I think we're going to move on.
Okay, great. Thanks, everyone.
So I guess the next sort of broad two-speaker theme was to sort of get at the question of functional interrogation of microbial communities, which is really sort of a critical area for research. And we're going to hear from Professor Jay Lennon on his work at Indiana University in this area. So I'll turn it over to you, Jay.